Okay, I'm beginning to record now, halfway through. So you see how it involves cloning, right? The zygote loses its nucleus and gains the nucleus of the mother-to-be. Now we have the product, which is a new zygote. The zygote has the nucleus of the mom-to-be and the cytoplasm of the donor. You see? Okay. And then that embryo is allowed to incubate in the lab, an average of what? How many days? 3 to 5 days, more or less, whatever it, just like in the natural process, it takes about 5 to 7 days to come down the tube and implant, right? So about 3 to 5 days, and at 5 days on average, they'll attempt the implantation. That's method one. Method two, both mother and donor go through the hyperovulation, but no fertilization yet. The nuclear swap is done before the eggs are fertilized, okay? Nuclear swap before, so the cloning is done before. And now the cloned human, in other words, an egg, not a zygote, but an egg with the nucleus of the mom-to-be, the pronucleus, and the cytoplasm of the donor, that gets fertilized, okay? And incubated 3 to 5 days, attempted implantation. Follow? Okay. Again, you'll get the diagrams and everything, so stay with the big picture for now. So those are the two methods. Now we're ready to do the bioethical analysis. Let's look at method one. Here's the mother-to-be, the father-to-be, and the donor. So in method one, both women undergo hyperovulation. So these are all the grievances, if you will, the reasons why not undergo three-parent embryo, okay? 2 women are hyperovulated. What's the issue with hyperovulation, ethically speaking? Right, the ovaries can burst, so she's risking her health and her life, right?
The other eggs.
There'll be spare eggs that we'll deal with in a minute, but just concentrate on the health of, so this is about the health of the mother. The mother has mitochondrial disease. Now we're going to risk her health to produce these eggs. Now, is reproduction a vital function? Yes and no. Is it vital for the spouses or the mom and the dad? It's not vital for them. You know, people can survive without reproducing, right? The individual is vital for the next generation. So it's vital at the population level, if you will. As long as some people in the population are procreating, then the species continues. So it's vital for the next generation, but not individually. In other words, I, as a mitochondrial woman, don't need to procreate in order to continue living, right? In fact, automatically having mitochondrial disease, I'm high risk for pregnancy. Pregnancy, do you think pregnancy kind of takes some energy out of the mom? Yeah, for 9 months, right? Here she is weakened with her energy because her mitochondria are compromised. And so good advice actually is it's not good for you to be pregnant. It's you're risking your health and possibly your life. So, you know, this woman undergoing hyperovulation, to what purpose? To have the child, right? Just as having issue. The donor is hyperovulating, so she's risking her life. Could she be getting paid for this? That sounds pretty unethical. Now you're buying eggs, you know, so you're trafficking with human cells here, with human gametes. Why would the donor, let's say the donor is a cousin or someone like that, and she just wants to help her cousin, all right? She's doing it for free. All right, but that's a big issue there. But it doesn't stop there. It involves at least 2 steps of IVF, but the first one is the father is actually fertilizing the egg of his wife and the egg of a donor. So this is kind of cellular adultery going on here. This father is fertilizing eggs of his wife and the egg of this donor. Granted, it's done in vitro, and that's the other thing about in vitro. You have procreation without the unitive aspect of it, which is also questionable. Certainly not nature's way, right? At any rate, now we have a donor embryo that is enucleated. So when we do that, essentially we kill that embryo. That was a human embryo. Okay, from two healthy human beings. And it's done this way precisely because they are healthy. So the moment we enucleate this zygote, we have killed that zygote. All right, we're only interested in his or her cytoplasm. Next, the mother's embryo, the nucleus goes into the cytoplasm of this donor, right? Essentially the cloning that I described. So now we're doing human cloning here. Okay. Attempted implantation. My point with attempted implantation, do you think that this is done with one egg of the mother and one egg of the donor and one sperm of the father, this process? No way, right? This is aside from being a boutique industry, catering to those who can afford it, it's also a shotgun industry, right? It's like hunting for quail. You use a shotgun and maybe a few of the pellets hit the quail. So it's got to go through several trials. Let's go back to Dali for a moment. Looked into Dali because that's when I went for my doctorate in genetics at Purdue. How many trials did it take to manufacture Dali? Anyone looked it up ever? It took about 250 trials to get Dali, all right? Trial and error. In other words, failed attempts. I think it was actually 258, something like that, over 250. Then they did a cat. Anyone remember Copycat? No? Copycat, it was the first clone cat. So what happens is after Dali, which was the proof of principle that mammals can be cloned, essentially the technique for one mammal is the same as cloning another mammal in the lab, pretty much, all right? In principle, the same technique. So they started cloning all kinds of mammals, and one big one, of course, was mice, transgenic mice, because you can do a lot of research with mice and transgenic mice, so they were cloning mice and so forth. And it was interesting that it's coming out at a trial of about 250. It takes about 250 more or less for whatever mammal it was. The dog took longer. And it was intriguing why it was taking longer for the dog. It turns out that the female dog, her ovulation is very mysterious. It's very difficult to identify when the female dog is actually ovulating. They're having a hard time getting mature eggs from the female, which, by the way, this is scientific. You can look it up if you would like. The female dog, you know the name for the female dog? It's called a bitch. And that's a scientific name. Well, that's the actual name for the female dog. That's why it's offensive to call the woman that, because she's not a dog, right? But that's the actual name for a female dog. Anyway, it was difficult getting eggs, mature eggs from the female dog, and that's why it took longer to clone. But now it's cloned, and it's in the pet industry. And if your pet dog dies, you can clone your dog now. All right? At a price, of course, of several $1,000s. Yeah. It's getting very weird out there. But if there's money, you follow the money. The technology allows it, money follows it, it moves the economy, and there's a price. Anyway, so it's a trial and industry. That's why it's going to involve dozens, if not hundreds, of these embryos to get one to actually implant and take and go forward. And then last the whole 9 months in the pregnancy, right? Because this woman, the mother, is already compromised because of her mitochondrial disease. So, in other words, it's really forcing nature galore here, all right? Has anyone stopped to ask this embryo, did he or she want to go through all that process and have all these dozens of siblings being killed so that he or she could have life. So these are the bioethical questions that sometimes people don't consider, all right? That's method one. How about method two? All right, well, the good thing about method two is that father is only fertilizing his wife, maybe. He's not fertilizing the donor eggs. So, okay, the two women still have to go through hyperovulation because, again, being a shotgun industry, we want to have a lot of these eggs to experiment with, all right? So the same risk to these two women is up there, up front. Then IVF process to obtain those eggs, do the nuclear swap. So again, nuclear human cloning here with what right? There are actually some laws that were passed at least at state levels to not do human cloning, meaning reproductive cloning. And this is precisely reproductive cloning because they wanted to, these scientists wanted to get permission for therapeutic cloning. Did we talk about cloning formally in class? No, okay, but you have reproductive cloning and therapeutic cloning. This is actually reproductive cloning. Manufacturing, doing cloning for the purpose of generating a new human being, reproductive, for the purpose of reproduction. Therapeutic cloning is doing human cloning just to have zygotes go up to the blastocyst stage, but never implant them, just to have a source of embryonic stem cells. Where do the embryonic stem cells come from? They come from the ICM. What is the ICM? Inner cell mass is what becomes the embryo proper, right? At what stage of the embryo? Blastocyst. The blastocyst, right? With the first cellular differentiation, remember after Morilla, the embryo hollows out, and then that little cluster of cells on the inside, the ICM, the inner cell mass, becomes the embryo proper. Those are the embryonic stem cells. So to obtain embryonic stem cells, that little embryo has to be disaggregated. We say in lab lingo, that means disaggregate means dissociate the cells, disassociate the cells, go after the ICM and put it in a dish by itself and grow a confluence, grow a cell line from there. So what does that do to the embryo when we generate embryonic stem cells? We kill the embryo. It's the ICM that is being destroyed. That's the embryo. So embryonic stem cell research involves killing embryos. When it's done in mice is one thing. When it's done in humans is another thing. We may kill mice. May we kill mice? Yes, if it's going to save humanity from some diseases, we do experiments with mice. They have to be justified, but we can. Should we do it with the embryo? Where is the embryo consent? The human embryo, right? So just a little side, I gave you human cloning in 2 minutes there. So the embryonic stem cells, human embryonic stem cells, already embryonic stem cells, come from the ICM of whatever species, and therefore it kills that embryo. And that's why we are against embryonic stem cells, because of the killing of the embryo again. All right, but you see, that's called therapeutic cloning, that kind of cloning. It's because it's from those stem cells, the scientists want to grow cell lines. How many cell lines do we have in the body, by the way? 2? No, that's the very first differentiation. You think of the blastocyst, okay? Fast forward to the live baby, 9 months later. All the organs in place, small. About 220 cell lines, about 220 cell lines. They make up our different organ systems of the body, right? About 220 cell lines. And that's the gold standard. That's what the whole thing about embryonic stem cells, or adult stem cells for that matter, that's the whole thing, to try to generate these cell lines in the lab so that the different organ tissues can be replaced when they are traumatized or diseased and so forth. That's the whole point of the stem cells, all right? Big megabucks, billions of dollars in stem cell research, embryonic and adult. Thanks be to God, the embryonic are not working. They're generating tumors because they have too much potential, and it's like implanting, they grow cancer. The adult stem cells are working. We'll talk about that later. At any rate, so you see there's human cloning involved here, all right? And that's exactly what it is. So we have to call it by its name. Whereas, again, the unethical way of obtaining that sperm from the husband, which is so broad in society today, but it's the truth. Second step of cloning, of in vitro fertilization, is when so the first step was to obtain the hyperovulation is the first step. The second step is fertilizing that, that egg, all right? And then the attempted implantations. So all these steps can be questioned ethically, and where's the justification? Has the embryo been consulted in all this? Absolutely not, right? Again, the husband/father to be here gets away scot-free. All the burden ends up always on the women, all right? All the burden is on the women. Oh, did I jump? I think I skipped. I was doing method 1, right?
No.
Oh, method, okay. All right, so method 2, again, the hyperovulation, the IVS, there's 2 IVFs involved. First, to obtain the eggs. Human cloning also occurs. Husband masturbating. The second attempt, the second IVF engagement is to fertilize the cloned eggs. And then the attempted implantation also, which is trial and error. And so all of these injustices are built in there time and again. Finally, I want to mention this. To what purpose? 3, oh, and by the way, this is 3-parent embryo, right? But it can also be 4-parent embryo. In other words, I said that the mother is compromised with her DNA, with her mitochondria, right? She has mitochondrial disease. That's the whole justification for this process. However, oh, okay, so perhaps a pregnancy for her could be dangerous. She's high risk automatically, and it's going to take a lot of ATPs and so on and so forth. So why not implant this embryo into a third woman who will be the gestation mother? Okay, and yes, you know, this appeals to our sense of we shouldn't go there, but it's happening. It's happening. It's they call it vulgarly rent-a-womb, but the women who will do this, not here in the United States, yes, for a few $1,000s. In Latin America or in Asia, they'll do it for a few $100s. There's a case in Singapore. A couple from Australia did in vitro, went to Singapore, and they just hired a young lady who was selling candy in the streets, you know, and she would make me maybe $100 a year in 2 years' time. Well, they gave her several $100s to gestate the baby. It turns out that they implanted 2 actually took, and but one had they detected a genetic defect, was Down syndrome, and so the couple who was paying for the process wanted to do, what do you call it? Selective reduction, get rid of the Down syndrome child. Well, the gestating mom said, no, these are mine. I'm gestating both. I'm not killing any. Okay, it was a big lawsuit, horrible. And it was international because the couple is from Australia, the young lady is from Singapore, and so they were trafficking. The embryos were sent abroad to be fertilized in Singapore, to be implanted in Singapore. So they're trafficking with human beings across international borders. It became a huge scandal. You can look it up in the BBC. Incredible, all right? At the end of the day, 9 months later, the couple walked away with their healthy baby, and the single mom kept her Down syndrome child. But then proceeded to sue the Australian couple for several $1 million. So now she has guaranteed health for her child. But this is the kind of stuff that is going on. This is the kind of stuff that's going on, all right? And so you see that these embryos could have then 3 moms. They have the nuclear mom, they have the cytoplastic mom, and they could have a gestational mom. Who is the mom? Will the real mom stand up? I mean, this kid could have 4 parents, 3 mothers and 1 father. I hope you see an injustice in that, a natural injustice to this child, okay, which is before any legal issues. We have natural rights. We have a natural right to breathe the air. All right, we have a natural right to have 1 mother and 1 father. Unless we're consulted, and we cannot possibly consent to any of this while we're embryos. So we have to go to the default thing, which is the natural thing. Okay, so this is called natural natural bioethics, if you will. No God, religion, or faith mentioned here. It's just nature's way. All right. So for all these reasons, we should stay away from 3 parent embryos. Just like we should stay away from in vitro fertilization. You mark my words. I forget who was the inventor of this, but it's in the UK. And mark my words, these inventors will come up for the Nobel Prize in Medicine. Just like Edwards and Steptoe were given the Nobel Prize in Medicine a few years ago for inventing in vitro way back in the 1970s, okay, 79. All right, let's end on a happy note. How about fetal surgery? This was actually, I was doing my first dissertation I was doing my first doctorate in Rome in moral theology and bioethics precisely. This was science fiction back then, okay? It was just a possibility, but no one was doing it yet. So I wrote my part of my dissertation was on this prenatal diagnosis, which is just coming out in the 90s and early 90s. And the possibility of fetal surgery, all right, back then. And so if we can do surgery on a human fetus, that fetus becomes a patient. So my dissertation was on the fetus, the human fetus, as a patient, as a medical patient, right? And all medical patients have certain rights. So that was my angle. At any rate, we can look at fetal surgery, which is a reality today, 20 years later. It's a growing branch of maternal fetal medicine, which is a big, big area in medicine today, all right? It's a broad range of therapies and surgeries that can be done today. It's an increasing field. It's very exciting. It's done in utero, and from one perspective, I'll tell you the punchline for this is that from one perspective, several perspectives, the fetal is an ideal patient. The fetus is an ideal patient, all right? You'll see what I mean in a minute. May or may not involve a hysterotomy, meaning surgery on the womb itself. In fact, one time I saw an actual womb excised, surgery on the fetus, and then the womb reattached and continue. The human womb, yes. All right, it was just fascinating. All right, so there are 3 main types of fetal surgery and therapies going on today: percutaneous, fetoscopic, and open fetal surgery. All right, so let's go through these. Again, you have them on the handout there somewhere. Percutaneous, these are some indications for it. Obviously, we're dealing with a fetus who has some kind of defect, structural or functional, and these can be detected either by sonogram or alpha-fetoprotein or other proteins that go through the placenta into the mom's circulation. So the mother's blood during pregnancy, at different stages of pregnancy, is a well of information. In fact, whole cells, now they've discovered fetal cells in the mother's circulatory system, all right? Very few, but real there. And of course, maternal cells also get to go into the fetus during pregnancy. So it's amazing what is being discovered of the interaction that is happening here. So what are we looking for? We're looking for the least invasive way of diagnosing the fetus. And if we can find fetal cells in the maternal bloodstream, then that's excellent because we can do karyotyping and so on and so forth on those fetal cells, right? If not, then we have to do a little bit of invasion on the fetus. It can either be by amniocentesis, which is going, sucking out a little bit of the amniotic fluid. There has to be enough fluid there so that the fetus himself is not damaged, or the embryo, right? That has a slightly increase in causing a spontaneous abortion or a miscarriage. I think it's about a 5% increase, at least. This goes back, don't quote me on this one, it's back to when I was doing my research on this, okay? So it's old numbers. It may be, amniocentesis may be a little, maybe better today. Let me write those names. Have you heard of these techniques? I mentioned them: chorionic villi sampling and amniocentesis. Let's see, so diagnostic tools, fetal diagnostic tools or techniques. Of course, sonogram is the best, non-invasive, right? That we know of. There's sound waves going through the womb and projected on a screen. Sonogram, of course, mother blood. Actually, this is fetal cells or factors. Typically, what are factors? Factors are typically proteins, right? But any chemicals, organic chemicals, typically proteins. Then this couple are non-invasive, at least to the fetus. Minimally invasive to the mom is essentially a blood test. But for the more invasive ones is the amniocentesis. So I think has about 5% higher risk of spontaneous abortion or miscarriage. And then there's another one, chorionic. Villus sampling. That's a little higher because that is actually taking some samples of the embryonic tissue or the fetal tissue, the chorion, which is one of the membranes, right? The amnion, the chorion, these are all membranes of the fetus around near the placenta here with a very fine needle. They got a few samples of villi with little hair-like cells, structures. And that, I think, carries about a 10% increase in spontaneous abortion or miscarriage for the mom. So it has to be justified, for great reason. But what if there is, of course, the first thing is there is a history. The first thing that is looked at is the mom's history, right? Her medical history, if she has an indication for something, all right? That could be inherited or something like that. You get it? All right. So if there is some indication of some abnormality, then it's follow up with some kind of testing, and any of these could be discovered, or it could be de novo. In other words, it's coming out in the fetus for the first time. The mom was totally healthy, and so was the dad, but the fetus is showing some kind of abnormality. So with fetal therapy, any number of these, it can be alleviated, and in some cases, totally eliminated, all right? Exciting. And you see it's minimally invasive. The other one is a little more invasive, but still considered minimal, which is when a cannula is introduced through a fetoscope. A fetoscope is just a scope that goes into the fetal area, and that's why it's called fetoscope. Otherwise, it'd just be a scope, which is essentially a rod, steel rod, all sterilized, of course, and everything else, thin, and that punctures into the amniotic cavity. And then through that rod, a number of things can be introduced, cannulas, and one of those cannulas could be a light, another cannula could be a laser fiber. And with the laser, some cauterization can be done. What's cauterization? Burning, right? Sealing off. When you have this twin-to-twin transfusion, which happens with twins when they have a common placenta, depending on when the twinning occurred. We talked about twinning earlier, right? Did we talk about twinning? Yeah. Monozygotic twinning, right? Homozygous twinning. Okay, so if they share a common placenta, then they're also going to be sharing capillaries, and typically one of the twins is stronger than the other and is going to grow stronger and can kind of cannibalize on the other, or the smaller one is non-viable, and either way you want to cauterize those common arteries and veins so that each twin has essentially its own placental supply and not be stealing nutrients from each other, all right? Because then one twin will tend to predominate and start cannibalizing the nutrients from the other twin. So it's called twin-to-twin transfusion syndrome, all right, which is, again, a range. When you see syndrome, it's a range of symptoms that occur. Some of that can be alleviated and in times cured with fetus-topic surgery. Finally, we're increasing in risk here, of course, but in possibilities. Full-blown fetal surgery on the fetus himself or herself, all right? And this involves that famous hysterotomy, which is opening the womb, opening the uterus, pregnant uterus, to do surgery within on the fetus. Here you can see an example. This is most likely a teratoma, which is a growth of tissue, fetal tissue. Sometimes it has teeth, eyes, parts of a human, but it's not a human, all right? These are teratomas. Here you can see a huge one bulging out from essentially the rump, the backside of this little fetus, who is head down, right? The rest of the body is inside the womb still. And so only this area is projected out, and they're about to perform surgery, which is a relatively minor surgery. As impressive as this looks, to disconnect this teratoma, or this bulge, this mass of tissue that can also be cannibalizing on the nutrients and impeding, even physiologically, just physically by being inside the womb. Imagine, much larger than the fetus himself, all right? Fetal surgery. There are so many pictures of this now on the internet, since I still have a couple minutes. Because a few years after I wrote my dissertation, the new examples started coming out, and there was a very famous photograph that you may have seen around of a tiny fetal hand holding the finger of a surgeon. Ever seen that photograph? It became very famous. And then since then, every surgeon that is going to do one of these surgeries does that. You know, they put out the little pinky at the end of the surgery, and the fetus will grab their pinky finger. But something like this, more or less. Here's the surgeon, all right? Here's the womb. Here's the hysterotomy. And there is the fetus holding on to the hand of the surgeon. Several surgeons, there's another hand here. Amazing, huh? So, yeah. Anyway, so from one perspective, why is a fetus an ideal patient? Think of a patient, think of a born patient. Let's say we got into an accident, God forbid, and now we're at one of the hospitals in town. Oh boy. Let's say that we're in intensive care. It was a bad car accident, all right? We were okay, but a drunk driver hit us. We were helicoptered into Jackson Trauma Center, all falling apart. We go into the intensive care unit, right? $10,000, $20,000 every day, maybe more. All the high-tech intervention, all kinds of monitors going on. Beep, 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 beep. We don't know. We're in a comatose stage. Maybe they save us, maybe they don't. All kinds of emergency. We got punctured everything inside. The cost is astronomical. And hooked up with all kinds of tubes. Got to be fed, got to be intravenous, got to monitor the vitals, you name it. All right. Here's a little fetus inside the womb. Is the ideal ICU, is the ideal NICU. Well, before natal, prenatal, PICU, this is the PICU, prenatal ICU. No cost, all right, sterile environment, fed by the mother, exactly what the fetus needs. Tiny, tiny incisions, which can be done today with, I mean, if we can manufacture an embryo, if we can put a sperm inside an egg, if we can put a sperm nucleus inside an egg, all right, with macromanipulators in a sterile environment, in a lab, we can do this. So this tissue is pristine. The cells are very young, you know, it's young tissue. It's going to recover quickly, maybe not even a scar by the time the baby is born. Compare all that to a born person like you and I, or even a child in an emergency unit in a hospital. So from many perspectives, the fetus is an ideal patient. We just need to work small. And know what we're doing. The catheters are all tiny, all right? But it can be done. It's a growing field. I think it's exciting, and it's something very positive. And it also calls the question, if the fetus is a patient, then HIPAA and everything else has to apply. Because now the fetus is at least partially outside of the mom. The head is not out, but how can we not consider this a human being? You know, what are we operating on, a palm tree? What? So it's so obvious. The issue is obvious, and that's why I chose this topic before it could be done, but I heard about the possibilities. I said, bingo, the fetus as a medical patient, right? I think it's worthwhile, and it's expanding, it's growing, it's exciting, but of course it's for high risk because it's high risk surgery, all right? But as the techniques make it better and so forth, to me this is good medicine. This is good application of technology, all right? And it's very respectful of two patients, the mother and her child. Okay. So I end here. I don't know if you have questions, comments, but this is basically beginning of life issues, a lot of controversial issues here, right? Now, starting Monday, we go to the opposite end of life, the end of life, and pick up some more controversial issues there. And then later on, we'll go into the middle of life with other healthcare issues. Okay, if you now have any questions or comments, shut this down and send it to you. And then you can send me your summaries by Sunday, right? Okay, thanks a lot.
Thank
You people have a good weekend. Thanks. Okay. Sorry? Yeah, oh yes, okay, so you're back, hold on, don't go. Eric. And, no.
I owe you.
Yes.
I completely forgot.
That's all right, no problem.
Thank you.
You're welcome. Immediate recall, push it into long term. Okay, let me see if I can stop this.